And now for something completely different, right? I'm going to tell you a little bit about a big project that Lee Hood and I have started called the 100K Wellness Project. And I just want to give you a little bit of the vision for this and then hopefully have a little time at the end to talk about ways that there might be some interesting interfaces. So first, just by way of disclosure, Lee Hood, Clayton Lewis, and I have started a company together. We're the three founders of a company called Aravale, and which is actually here in this building. I noticed as I walk in, there's a big poster saying "Welcome Aravale Pioneers" for people that sign up for this. But just to make this clear, Aravale is a partial funder of the project that I'm going to talk about today, our pilot study, which we call the 100-Person Wellness Project. Okay. So this is Larry Smarr, and I don't know how many people might know who he is or not. Probably not too many in this audience. Larry is what you might consider either your dream or nightmare version of a patient. He has become, I guess, not the poster child, but let's say the poster man for the quantified self movement. Larry is the head of the San Diego Supercomputing Center and has been measuring an immense number of things about his body for the last 15 years or so. And in fact, I was just with Larry last week when I gave a talk down at UCSD, and he has a set of monitors bigger than, let's say, the entire wall here that just zooms in and shows data that he has about his body dynamically at any point in time. And so Larry had an interesting experience a few years ago, which is he had been monitoring a large number of parameters in his body over a period of time, and all of a sudden he has this huge event where a number of these parameters spike to levels that are 50 to 100 times, in some cases, larger than what he's ever seen before. So he goes into his doctor and he says, I've done an analysis of this data, and he says, I think I'm getting inflammatory bowel disease. And his doctor says, well, how do you feel? He says, I feel fine. He says, well, there's not really anything we can do for you at this point in time. So he goes, he gets the same response from a number of people. And anyway, what happens is that a few months later, he has an acute episode, ends up having to go to the emergency room, and sure enough, it turned out that he, in fact, did have inflammatory bowel disease. Now, that's just an anecdote, right? It's an N of 1, as we went through a lot of N of 1 interesting case studies this morning. But basically, what we want to know is this an experience that is generalizable to a lot of people? How much warning signs can we find in our body if we were measuring that would tell us before a big event happens that it's coming? Okay, so that's an anecdote. So what we want to do is take this experience of Larry's and translate that into large numbers of people so we can do appropriate statistics, so we can build appropriate systems to try to see to what extent can we predict disease before it happens. So of course we have an aging population. This is not a problem, right? This is what we wanted to do. This is the point of vaccines, it's the point of antibiotics, the point of clean water, and all the things that our society has done. But we do have a population that's rising in age, as everyone in here would be well aware of. And as a result, we also have what is a problem, which is that there's a lot of chronic disease, and that is also on the rise. It is estimated by the CDC that 86% of healthcare costs treat chronic disease, so there's a major economic driver here. And so what we would see, or setting up the problem, is that most of our current medical field is focused on care after illness, right? And there's all kinds of reasons for that that are practical, but for the most part, we're not set up to deal with the kind of thing that Larry comes in with, an analysis of data, right? We wait mostly for symptoms. Data in general is based on population averages, not deeply on individuals. I will say that I think that there are certainly pockets of where this is not true, and I think what we saw this morning was a beautiful example of diving into individuals. And I think part of that is driven by the great imaging technologies that you've got, because you can see very plainly, what's happening in an individual. We want to open up data to make that possible in a wider number of fields. And then the third issue here is that we don't really know in any great amount of detail when we might become chronically ill. And so the question is, what if this were different? And could we think about ways to try to accelerate making that different? So in every person's body, there is happening at this moment in time aspects that are either driving us towards greater wellness or greater disease, and we might have these things that are happening in our body that lead to particular futures that we might not, that we won't want or aren't interested in. And of course, we know that we don't just want to be able to predict these things, we really want to be able to stop them. So how do we go from being able to do prediction to prevention? And so what we think about here then is wanting to understand better how the systems of our body are supposed to perform optimally. And it's interesting, I was on a panel recently with Denny Ausiello, who I don't know if you know him, he's the former chairman of Harvard Medical School. And so we were on a panel together, and he made a comment there following this talk that I thought was really interesting. And he said, medicine is the only discipline that doesn't study its own gold standard, which is wellness. And I thought that was really an interesting take because we are trained so much to think about disease and to think about medicine in the concept, in the paradigm of disease. So if we look at determinants of health in the United States, 30% of a person's health over the course of their lifetime is attributable to genetics, 60% is attributable to behavior and environment, and it's estimated that about 10% is related to healthcare. Now that has a huge impact, obviously, in those acute moments when this is really important. But over the course of someone's life, there's a lot of other factors. And so what we want to do is to tap in more to that genetics and behavior and environment aspects of people's health. So one of the things that we're very interested in is launching what we call a new sector, which is scientific wellness. And so, as everyone in here will be very aware of, U.S. healthcare spending is about $4 trillion. And so what we see happening is, and there's a wellness industry currently, but that wellness industry is plagued by a number of issues. Sometimes there are a lot of claims out there that don't have a lot of scientific basis to them, or are not tied as strongly as we would like to scientific outcomes. So what we see is the rise of a field that we'll call scientific wellness. And the main idea of scientific wellness is just to take a huge amount of data that we can now measure about our bodies, which we couldn't before, and to try to use that to optimize wellness and minimize chance of disease proactively rather than, say, waiting for symptoms to occur and try to fix it after the fact. So we see the rise of this new industry coming. And it's one of the things that we're talking a lot with people here about because we believe that Seattle could absolutely be the epicenter of this new industry. So these are, of course, very challenging things. The grand challenge in biology and medicine is really complexity. This is a slide Lee likes to use a lot of this notion that there are going to be a virtual cloud of billions of data points around every one of us. We can aggregate and utilize a lot of new technologies for systems medicine so that we can understand how biological networks are connected and how they get perturbed in disease, and use that as an organizing principle to handle all of this big data. So one of the paradigms that Lee Hood, who's the president of our institute, as we mentioned, has really pioneered is a concept of medicine called P4 medicine, which stands for being personalized to the individual, of course, which is familiar, predictive, preventive, and participatory. And participatory is really an interesting aspect for us because we think about how do you empower patients with knowledge so that they can be right at the center of optimizing their own health. So in terms of conceptual themes then, we think about two major divisions, quantifying wellness and demystifying disease, and these being the basis for the wellness industry and the disease industry. And so to make that a little more concrete, right, because we're just sort of talked about a lot of big concepts so far, Lee and I launched together a project that we called the 100K Wellness Project. And this was, it's Lee Hood, so it was a little bit grandiose, but basically what we decided to do, or what we announced to the world, was that we were going to recruit 100,000 individuals, do whole genome sequencing on all of them, and then longitudinally over time measure proteomes, metabolomes, clinical chemistries, wearable devices, and so forth, with the goal of building an enormous database of with molecular detail that we could mine to look for the early warning signs for the major human diseases. And this was the initial goal of this project. Okay, so that's a very big project. If you, when we were first talking about this, just a back of the envelope calculation in your head, you say, okay, what would that cost? And the answer is about $10 billion. And so you'd say, well, that's quite a large amount, which it is. Lee being Lee, of course, lobbied Congress for the $10 billion. Shockingly, they didn't give it to us. But anyway, that led us to think about how do we do this in a different way. And so this led us to found the company Aerovale, which is now enabling us to scale this. And we actually think we'll go way beyond the initial vision of the 100K now. So I already mentioned the founding of Aerovale. In the interest of time, I'm not going to go through this, but suffice it to say we've put together just a fantastically amazing team. And it's not just about the science, it's also about the behavioral impact that you have for individuals who are optimizing their wellness, teaming them up with coaches and so forth, and then having all this then feed back into the healthcare system, which really has to deliver on treatments for individuals. And we've raised just under $40 million for this so far. So we launched in May of this year. We've got 70 employees now. We've got 1,000 people have come through the program. We'll be to 10,000 by the end of the year, and then we'll go nationwide next year. So there's a big impetus here. Okay, so what we wanted to do then was to take 100 people, and that's what we did in 2014, through this program so that we could prototype and see, was there enough information by getting this kind of data that would have an impact on people's lives? And so the reason for doing this is that one of the things that I'll argue is that we often don't study what we really want to know in health and disease. And I'll use myself as an example. So I have a grant from the NIH on Alzheimer's disease, amongst a number of others. But basically, we study the brains of people who have Alzheimer's and who don't. But when do we do that? Right after they're dead. Right? No one's given you a piece of their brain before then. And so in thinking about that, though, what we really want to know is 20 years before, what was happening in the body of one individual that led them down the path to Alzheimer's compared to the person who didn't. And what we really, really want to know is if we had been able to see those signs, could we have stopped that path? Right? And so those are the kind of things that we're really interested in building this big longitudinal study. I'm going to skip this in the interest of time. Okay, so we launched what we called then the Pioneer 100. It says 100 participants here, but it's actually 108 participants. A few people wanted to get in at the end that we accommodated. So they were mostly here in Seattle, not 100%, but mostly. Age ranged from the 20s all the way to 88 plus. It was a 9-month study run under an IRB, so we had about a 40-page set of regulations to the IRB that we had to follow. And then we tried to give people insights for the next phase. So this was whole genome sequencing, detailed blood, urine, saliva measurements 3 times a year, gut microbiome 3 times a year, continual self-tracking and lifestyle monitoring, mostly just via Fitbits and things like that, data integration and correlations. We had monthly coaching sessions for people so that we could make the data as actionable to them as possible and then do in discovery. So what this means at the end of the day is that we have millions of data points for every individual. We also have some discovery assays, metabolomics and proteomics and so forth for everyone. And what this is enabling us to do is just build a big data cloud around every individual. And then this is filtered back through the wellness coach. And we also had a study physician, Craig Kleebler, who reviewed all of the cases. And so whenever we come into something that was that looked like it was a disease, all that gets referred back into the healthcare system so that it's appropriately dealt with, and then they make the diagnosis and the treatment. Okay, so what we're building then is a research portal. So this research portal will have a ton of data in it, which is breadth. It's going to have a lot of frequency. We're going to be monitoring over time, so it has depth. And then we also have the impact of the recommendations. So because we have weekly contact with these individuals, monthly calls, weekly emails, we're able to identify a number of factors that might be leading to changes that are observed in the data. And then the whole goal of this is to build out scientifically validated metrics for us to understand optimization of wellness as well as what are early warning signs for disease. So by going through this then, what we're able to do, and this is just from the first 100, and this is for a paper that that's under review right now. And in essence, each of these blue lines here is a correlation that has been found that survives multiple hypotheses that links something in your microbiome to something that's changing in the proteins in your blood, or something, the metabolites in your blood that are changing as a function of your genetic traits and so forth. And I'll give a couple of examples of this. What I want you to contemplate just for a moment, though, is this figure, because no one has ever measured all of these variables from the same set of humans before. So a lot of these correlations have never been investigated in any way before. And so this is just from the first 100. We've already got 1,000. We'll have 10,000. This is going to get more and more complex. And these are all basically simple correlation measures using something called Spearman's correlation, which is less prone to outliers because it's a rank-based measure. But anyway, without diving into that, this is a simple version of this. As this goes to 10,000, 100,000, and we hope ultimately to millions of people, this is going to enable us to build very complex models to understand relationships in systems in the body that we just have not been able to look at before at the molecular level. So just to show, so I'll just pull out 4 of those correlations just to give you a sense here really quick, and then I want to wrap up so we have at least a few minutes for questions. So on the top here are a couple of correlations. I don't know if this is, and basically just positive controls. So these just say that if we have a predictor for, say, a genetic trait. Okay, so out of the literature, you can pull, we can build models that will predict for an individual, if all we know is your genome, we can predict, for example, the bilirubin level that's likely to be in your blood. Okay? And we compare that to the actual bilirubin level, and you can see that we get pretty good correlation. In that case, 0.48. That's just essentially a positive control. That's about how good you can get. So this shows that when we pull data out of papers, we build models, we apply them in our 100, it holds up. Now, what's really interesting here are some of the new correlations. And I'm just, in the interest of time, going to talk about one of those, which is this one here. So what we find is that when we have a genetic predictor for, in this case, inflammatory bowel disease, what we find is a very strong correlation. Again, you can see that 0.48, the p-value, 3 times 10 to the minus 13th. Very unlikely this would happen by chance. But what we find is that people who are at higher risk for inflammatory bowel disease tend to have lower amounts of cystine, which is a dimer of cysteine, lower amounts of cystine in their blood. So why is that interesting? So what's really interesting about that is if you do case control studies, just the normal sort of case control for people that have inflammatory bowel disease and people that don't, it turns out that one of the top biomarkers that comes out of that is this level of cystine. Okay? Now what's interesting here is that we find that relationship in a population in which nobody has inflammatory bowel disease. We're just computing their genetic risk for the disease. Okay, so what that means is that we can use genetic risk for a disease as a phenotype on which to study aspects that are related to the lifetime chance of manifestation of that disease. So that opens up a very new paradigm for us. Second is that we can also use this kind of information as a correction on case control studies, because it popped up as this biomarker in this case control study, but what we find is that it may be that that was totally pre-existing, right? In other words, it has nothing to do with the onset of the disease. It just is a function of the fact that your cases were more genetically at risk for the disease they got than people who didn't, right? That stands to reason. And so because of that, we already know from this data that there are a number of differences baseline between individuals that are at higher risk and low risk. And then the third most tantalizing possibility, and then I'll wrap up and not finish out so we have a few minutes for questions, but the third most tantalizing possibility is that, and we need more evidence for this, is that there could be a link between this lower cystine and your lifetime risk for inflammatory bowel disease. And there's actually a plausible mechanism. Cystine sits upstream of glutathione pathways. Glutathione pathways, of course, are related to combating oxidative stress in your body. Oxidative stresses are a key driver for inflammatory bowel disease. So it might be, and this is where we would love to partner with groups like here at Swedish, because we want to launch clinical trials around this eventually. But what it might be is that you could is that this lower amount of cystine over the course of your life makes you more susceptible to this oxidative stress. That's why you get inflammatory bowel disease. If that were true, then what you could do is you would take something static, like I'm at high genetic risk for this disease, and you turn it into something that's trivially easy to do something about. Oh, and the driver for that is because you have this low cystine throughout all your life. So we're going to either alter your diet or we'll give you a supplement for cystine. And this is going to enable you to have better functioning of your glutathione pathways, which we can monitor in the proteomics data. And that will now make it so that you are at low risk for IBD throughout the course of your life because we understand how the genetics mediates through to the phenotype. Okay, and so that's really the key idea. And so there are a huge number of new possibilities of how we can figure out preventive medicine by going through this kind of data that was just totally not possible before. And so in the interest of time, I won't go through the rest of it, but let me just thank especially Lee Hood, who's been just a fantastic visionary leader and friend and mentor through all of this, and the amazing team we have put together, especially two former students of mine, I have to say, Andrew Majors and John Earls, who did almost all the calculations for those correlations that I showed. And with that, I'm happy to take any questions. Thanks.